Hi, everyone. Thanks for joining in. And Ron invited me to, tell you a little bit about education at the MBL. And so I'd like to tell you, what we think makes the education that we do here at the MBL somewhat unique. And let me sort of launch my slides here. Okay. So as Ron gave a nice introduction to a couple of the aspects of the MBL. And so, we're located here in Cape Cod, Massachusetts, right on the water. And as you heard, the MBL was founded in 1888 as a place that naturalists convened to study the biological world. And the reason that we're located where we are is that this is an unusual spot for a confluence of various currents. And it means that while our waters are not as pretty as some of those reefs you saw in Roger's movies, they're incredibly nutrient rich and incredibly great in biodiversity. And so that's one of the things that drew early researchers here in the late 1800s. But the people that came here and started doing research at the MBL, they also had a mission for education. And so during almost this entire history of the MBL, education and courses have been a really important component of what we do. And here's a picture from 1893 from one of those courses. And this tradition has continued and grown. And it's one of the things really that MBL is well known for. And it's one of the things that makes us incredibly crowded in the summertime. So during a typical sort of summer season, we'll offer these 21 courses right now. These are advanced training courses. And you can see that we've grouped them here into a couple of different categories. Now, some of these are extremely long running, like the physiology and embryology courses. Others are brand new, like the deep learning course that we're doing, which is this next summer will be the second time we've taught it. And these courses tend to be quite long. So many of them are 6 to 8 weeks and very intense experiences. So we end up with about 500 students, and they're mostly graduate students and postdocs that take these courses, these advanced training courses, but about 500 faculty rotate through and are involved in teaching these courses. And so we have scientists from more than 300 institutions from over 30 countries. Now, as Ron mentioned, these courses are characterized as being transformative. And if you get feedback from the students that take these courses, that really attests to that these are amazing experiences for these students, right? And you see that not only from the comments from the students, but the comments from the PIs that send their students and postdocs to these courses. And this, I think, is really a very true statement that, for many times a student comes here, but they go back a scientist, right? So what makes what we do special? And I'll say that, we're not totally unique, right? There are other institutions that also offer great advanced training courses. So I've been involved in teaching, for example, at Cold Spring Harbor as well. But there are a couple attributes, I think, to MBL that really are different and that make us so successful. And I'll give you a couple, but the first ones I'm going to start with are maybe, important, but maybe not the key. So there are things like we have available to us all sorts of cutting edge equipment. So this is just an example of some microscopes, things that get built here in the courses, and then things that are loaned to us by companies. We have an amazing loan program that allows students to use all sorts of state-of-the-art equipment when they're here. We have a wonderful building that's devoted to teaching, and all of the rooms in that building are really specially designed for teaching, for hands-on laboratory work. We also take great advantage of the fact that we are a marine lab. So almost all of the courses will actually use this diversity of organisms that we can collect from all different phyla because we're here in the ocean environment. We have a collecting vessel. We have a whole big building that's devoted to maintaining many of these organisms. We also have this long tradition then of having all of these people descend on us in the summer. So, as people like Roger and Carrie are here year round, and our resident science staff of about 25 labs, a total of about 125 people. But we grow enormously every summer. So we suddenly have 500 students, 500 faculty and TAs, and then a very important visiting scientist program of about 300 people that come in the summer that really add to this incredibly intense interactive environment. And so people like Ron, continue to come here every summer. There are people that have been coming for 30, 40 years to do research in the summertime and set up collaborations. So it's a great environment to network. It's a great environment to meet new people and to come up with new ideas and new collaborations. So I think all of those are important, but I'll tell you what I think is the real secret. What it is, it's the mindset that we have in the way that we teach these courses. So what's the real secret, or, you know, the special sauce or something that may be there? So I'll break it down to a couple things. One is really an incredible work ethic that we try to put onto the students when they're here. They know that they're going to be expected to work hard, but they have a great time doing it. And I have to say, there's nothing more pleasing than like being in the LURB teaching building at 2 in the morning and seeing how packed it is with people that are actually having a fun time doing science and really enjoying it. And I have to say, I've been doing it now for 20 years, and I still love that feeling of being involved in the teaching here. The other is that we really instill in the students an idea of being fearless in what they do. So a typical kind of module in our course, we're teaching students the techniques that they need to do a set of experiments, but then we rapidly sort of transition them to actually asking them to design their own experiments, but not to design something that we already know the answer to, or is really sort of a trivial application of a technique, but ask them to actually go out on a limb and try to do something hard, right? And to really be imaginative in what they do. And that we will do whatever we can as instructors, faculty, and TAs to help them work on what they want to work on. And I have to say, even though sometimes we think this is just totally ridiculous, there's no way this is going to work. But we're like, we never say that to students. We're like, okay, let's try this. Let's do it. Okay. And then the other thing I think we teach the students, and it's a kind of a strange thing to say, is we teach them to fail. So we sort of instill in them that if they're not failing, well, they're not really trying hard enough, right? So they fail, and then we teach them to fail some more, right? So to really go for it, to really be imaginative and creative, and then that's really the key. And of course, there may be a lot of failures, but when there's a success, it's often an amazing success. So what I wanted to do is just highlight some actual real life examples of how this has worked, and some things that I've been involved in, and some experiences I've had. So I'll reach back to 2015, and this is one of my good colleagues and friends, Ishwar Hariharan. And Ishwar was teaching in one of the courses, and he had given a lecture on the regenerative capacity of wing imaginal discs in Drosophila. So as you know, adult flies have wings, but there are these imaginal disc structures in the larvae that grow into the wings. And experiments done long ago with surgical experiments had shown that there was a capacity to regenerate. And Ishwar developed, and along with other labs, a genetic system for ablating the wing and being able to show that you could regenerate the wing proper from other parts of the imaginal disc. And he and other labs have gone on to show the genetic pathways involved in this regeneration process. So one of the people taking the course at the time was Julian Kimura. And Julian heard this lecture from Ishwar, and we were moving on in the arthropod in this particular module to working on other insects other than flies. And so my lab, one of the things, as Ron mentioned, is we work on butterfly development and patterning in butterflies and structural color. And so we had a bunch of caterpillars and chrysalises and everything, and we were having the students do some experiments looking at pattern formation. But largely the idea there was to work on fixed tissue. So for example, to dissect the imaginal discs out of this caterpillar and then to look at pattern formation that was going on. But Julian said, well, wait, I just heard this lecture from Ishwar about how imaginal discs, you can get this wing regeneration in imaginal discs in flies. I want to try this in caterpillars. And we had a technique where we could surgically make an incision in this caterpillar and the disc would pop out. It's a way we often harvest discs, and then we throw the caterpillar away. And Julian said, yeah, but I want to see if the wing will regenerate. And of course, in our own minds, we're thinking, oh my God, this is a waste of time because there's no way it's going to regenerate because there's nothing to regenerate from. But we're like, okay, Julian, we'll help you. And we get caterpillars and he does all these surgeries, gets all these animals, the wounds heal, and he failed. Okay, wings did not regenerate, but he got a fantastic result, which is incredibly scientifically insightful. So when these animals pupate, when he's actually removed the forewing disc, he gets a pupae that now not only is missing the forewing, but it's missing the covering over the remaining hind wing. So one of the problems that we'd had in studying butterfly wing development is that the chrysalis is opaque and we can't see into it. And any surgeries we do to make it visible usually lead to some sort of damage. But this technique allowed us to get pupae where we could see right into the hind wing. So what you're seeing now is a movie that's representing about 8 days of development. And we can see all of wing patterning going on, and scale growth, and nanostructures that make some of the structural color. And so Julian's failed experiment led to a great boom in the experiments we can do. And in fact, we've used this in subsequent years in the courses for the students to be able to do some cool experiments with wing development. So one of the things we've also been thinking about is, can we sort of take this on the road and have these kind of MBL courses in other places and help to seed those kinds of things. So this I'm showing you this is Roberto Mayor. Roberto is a faculty member at UCL in London. And Roberto took one of the courses about 35 years ago at the MBL. And he's one of these people that will tell you that it's a transformative experience. Roberto's from Chile, and he thought, well, I want to be able to make this kind of course available to students in Latin America. So a number of years ago, Roberto established a mini sort of one of these courses that very much in the vein of what we do at the MBL in Chile, that was open to students from all over Latin America. There are many of us that also teach in the MBL course that come and help him in the wintertime in Chile. And it's great fun, and it's a great example of being able to sort of take that same passion and spread it around. And again, it's at a marine lab, so we can take advantage of many of the same creatures. But in addition to these sort of advanced courses, one of the things that had occurred to me for many years is that it's kind of a shame that we have this incredible facility, these buildings and all of this equipment. And we are completely packed to the gills in the summertime, but we have a great capacity to also do teaching in the non-summer months. And so the MBL has been actively making efforts to actually expand that teaching mission. And recently we've had courses for secondary high school students, as well as for undergraduates. And so we just got done teaching a set of undergraduate courses, and the students just went home a couple of weeks ago. And one of the courses was one that I helped teach, was an imaging course. And we taught them some basics of imaging, running a microscope. So we showed them a confocal and said, okay, this is a $500,000 piece of equipment, but you have to use it. So you have to learn to use it and be confident in using it. And then towards the end of the course, again, we tried to get the students to come up with independent projects that we could help them with. So this is Michelangelo Neff. And Michelangelo is a second-year college student. He's a computer science major. And in fact, he's never taken any college biology. All his background was from high school, and he'd never held a pipette pen in his life. And so, we were thinking, okay, let's think of a really challenging set of experiments you can do. And one of the things that we talked about, and you've heard about a little bit from Carrie, are the Hox genes that you were originally discovered in flies. And as Carrie mentioned to you, these genes are organized along the length of the chromosome in the same order that they're expressed along the embryo. But what you always see in textbooks, and you even saw this then in Carrie's slides, are these nice cartoon images that show where these genes are expressed. But if you actually work on these Hox genes, it's actually sometimes a little annoying, because you know in detail that these images never quite right. So, and sometimes have some kind of hilarious mistakes, like this actual embryo has 4 thoracic segments instead of 3, which no insect has. So this gene expression pattern can't be correct here, right? And so what we did is we set up a challenge for Michelangelo and for me to help him with, which was to actually make a real image of an embryo showing the expression of all 8 Hox genes, right? So to do that in situ hybridization experiment that Carrie described, but to do actually 8 probes at once and to resolve them all. And now that's not an impossible experiment at all, right? But if I asked a postdoc or a grad student in my lab to do that, someone with a lot of experience, they'd probably say, that's going to take me a couple of weeks to troubleshoot, and it may not work, and this is going to be hard, and I'm not sure I want to do this. But of course, the great thing is that we could just work with Michelangelo, right? Very naive in some ways, right? And just present him with this challenge, and again, instill in him the idea to just be fearless with these experiments. And he got it to work. So we get this great image, which I show you here. So the fly embryo, and he's like hybridized for now all 8 Hox genes and been able to resolve all the in situ patterns beautifully. So now you have a real image of how these genes are actually expressed, right? A textbook sort of to replace the textbook drawings that you often see. And of course, we had 8 probes working. We said, well, let's push our luck a little bit. Let's go to 9. So we added in a gene called engrailed, which marks the boundary of the segments. Made it a little harder to do all of this imaging, but he still got it to work. So again, you can see here, he's done the in situs and the imaging. for all 8 genes. And so it's a great example of really just convincing someone to be fearless and to go for it. And of course, there are failures. So we tried to do 11 different probes in zebrafish and we failed. We only got 7 of them to work, but it still looks pretty nice. And we'll keep trying. And so I think it's a nice example, I hope I've given you, of the kind of inspiration we try to give the students to be fearless, to not be afraid to fail, to give them every bit of support that they can, and to give them the time in having these fairly long courses to be able to really have that confidence. And so with that, I'm happy to take any questions. Thank you very much.
Thank you so much, Nipam. That was a really fantastic introduction to education at the MBL. I think that the program you have there is very impressive and also very inspiring. I'm going to start with a quick sort of obvious question here is, how has the pandemic affected MBL's ability to offer these educational courses?
Yeah, thanks. Yeah, so obviously we had to not have any of our courses last summer, which was a very difficult thing to have to do, but I think everyone understands the need to do that. And so what we did was we went to some of remote programming, so we had lectures and things like that. But of course, what we're really excited is the actual in-lab, the intensity of that experience and the training that comes with it. So I'm happy to report that we're doing the planning now to be able to actually have and to socially distance and to have probably about more than half of our courses occur next summer. So we're doing all the planning that we can for that, lining up the testing and things like that. So that's definitely been, an issue for everybody. But we're looking forward to getting through this. We figure next summer we won't be completely back to normal, but we will be offering courses, and we're excited to have students here. And as I said, we had these undergraduates here, so we did that safely. So we know a little bit about now what we need to do to make this work.
That's great. So tell me a little bit about how the applicants for these courses are evaluated. What are you looking for in the students that join these courses?
Yeah, so each course is a little different in how they do this. So we try not to micromanage the courses. The courses have course directors, and they do a lot of this decision making. So usually a course has a small committee that's put together that reviews the applications. And so we're looking for students that are excited to really push boundaries. We look for that in things that they say in their application, that they realize the sort of level of commitment they're going to have to these courses. And we try to get a really diverse group of students. So for example, in something like, a course, we may actually, have great fun by bringing in some students who really come from an engineering or a physics background into a more biology oriented course so that they can contribute, learn, and exchange ideas.
So you mentioned that some courses have been running for a long time and others are brand new. How does the roster of courses that you offer sort of evolve over time? How are new courses, added on, and perhaps other courses, no longer offered?
Yeah. So we have an education staff here, but then we also have a group of people that form a committee they call the Advanced Training Course Committee. And they help us to routinely review the courses that are here. And, there are courses that after time, it may be time for them to, to wind down. And then that gives us opportunities to offer new courses. So right now, offering additional courses is always a challenge because we're so crowded in the summer. But we do have opportunities kind of in the shoulder months, earlier in the spring and in the fall. So there is some evolution of the courses. So we have had courses that have come and gone over years, and other courses that have lasted for incredible periods of time, 150 years or more.
Yeah, that makes sense. So obviously the MBL's educational programs, as was stated by the students, are very transformative and career changing. But how is the program as a whole, or even just the individual courses, evaluated? What are the sort of metrics of success for the program overall?
Yeah, so we often, or had for a long time, collected evaluations at the end of the course. But one of the new things that we're doing now is actually asking a series of questions to the students and to their PI advisors before they come to the course. and then asking a lot of those same questions after the students have gone back after the course to really evaluate how things have changed for them. And one of the things, and we've got some preliminary data already from 2 years now, showing that, you know, the students really do change, not only in the experience they have in terms of techniques and learning things like that, but in their confidence in doing experiments. And that's reflected both in the students' own comments and the comments from the labs they're coming from.
And how about the trajectories of the students that come in, especially like the undergrads and the grads? Do you guys ever track their career outcomes or what they end up doing?
Yeah, I'd say we in the past have not done a great job with that. So we're kind of in retrospect now going back and trying to track a lot of our students. But yeah, we're collecting information on the kind of things that they've done. And you know, I think if you go to any meeting in a lot of areas like neurobiology or cell biology or developmental biology, you'll see these giant reunions of people that have taken these classes getting together and talking about their experiences and how it shaped their careers.
Yeah, I think that's amazing. I think right now that's all the time that we have for questions, but I just want to, again, thank you, Nipam, for that fantastic overview of education at MBL. And of course, Carrie and Roger for their great presentation. I think all of it was very impressive and inspiring. And I have not yet been to MBL, but I look forward to coming to visit for sure. And I want to let the audience know that today's speakers have promised me that they will head over to our Slack channel after the talk just to answer any questions that you perhaps did not get answered. And there's an invitation in the chat box that you can click on. It will take you to the Slack channel. So thank you all again for joining us. Thank you to the audience for joining us. Life science across the globe is going to take a little bit of a hiatus for the holidays, which for many of us is winter and some of us are summer holidays. And we will resume on January 13th. So I hope to see everybody then. Thank you.